All right. So, last video we talked about how proteins are made and how the message is coded for in the messenger RNA. Okay, so again, it's by 3 letters at a time. Those 3-letter sections are called codons. And when you're reading this, we read, we need a pen. Okay, when you're reading it, take the first letter from here, the second letter is the column, and the third is over here. Okay. One thing that I didn't mention, which I meant to and I usually do, I want you to have a look at it right quick and tell me, okay, what amino acid is coded for by the letters A, C. Okay, so you have A. This is your first one, so we know it's in this row. You want the second one to be an A, you want the third to be a C, so there it is. So AAC is, I think it's asparagine, if I remember correctly, or arginine. Asparagine, maybe. No, arginine. Okay. If you look and tell me what amino acid is coded for by the letters AUG. This is a really important one. AUG. So we look, we know the first one is an A. And look in here. AUG, there it is right there. Codes for methionine, MET. This one you should know. Okay, it doesn't say in the chart. This is always going to be the start codon for the kinds that we're going to be doing. Okay, that is what you're going to be looking for in order, that's what the enzyme will look for in order to start coding or start translating the message. So that is what we're going to look for when I give you problems. Okay, so AUG is the start codon. It always codes for methionine, but because it doesn't say start in your chart, you need to know that is the one you're looking for, AUG. If you look, oh, I need to erase that. There are 3 codons that tell the enzyme to stop making the protein. Okay, so if you look, there's one, there's the second one, and there's the third one. So UAA, UAG, and UGA are all stop codons. So when the enzyme comes along to, or sorry, when the ribosome, what am I thinking? When the ribosome comes along and reads the stop codon, it knows to stop adding amino acids to the protein chain. Alright, so this video is going to focus mainly on the next process, which is translation. Okay, and like we saw before in the simplified diagram, translation involves messenger RNA, ribosomes, and amino acids. But one thing we didn't know about was this little guy. The little t stands for transfer RNA. Okay, this is kind of what it looks like, transfer RNA. So it's RNA, single-stranded, but it folds up on itself to make what they describe as a cloverleaf shape. Okay, within the cloverleaf shape, at the bottom here, is a three-letter anticodon. Okay, the anticodon pairs up with the 3 letters of the codon. So each codon is going to have a specific transfer RNA molecule that will bind and recognize it. When it does, it is carrying a certain amino acid, the amino acid that's coded for in the codon. So that is how it goes from the messenger RNA to bringing about or bringing along the proper amino acids. Okay, the anticodon at one end, right here at this bottom end, and the amino acid is at the other end of it. This picture down here is showing the ribosome. Okay, the big pink thing is the ribosome, and the ribosome is, remember, what attaches and reads the messenger RNA. Here's the messenger RNA, here's the start codon, AUG, and it's moving to the left through the ribosome. Okay, actually the ribosome moves along it. So, oops, I think there's stuff on there already. Eraser. There was. Not what I meant to do. Sorry. Okay, so the ribosome moves along the messenger RNA this way. If you look, here is, there's one that's going to code for methionine. There's two, oops, that would be another amino acid. So here's one amino acid, and here's the second amino acid. Okay, this is the incoming transfer RNA, so it's going to come in here and bind. If you look, it has the anticodon UUU, which will match up with the codon AAA. So if you remember, it's always C's and G's, and for DNA it'll be A's and T's, but for RNA it's going to be A and U. So the anticodon UUU is going to come and bind. So we know that this transfer RNA has to come and bind next. When it does, this amino acid will be joined onto the growing chain. Okay, so like the steps say, codon in messenger RNA is read by the ribosome. Transfer RNA with the proper anticodon binds. A peptide bond, which we learned about last year, but you may have forgotten. Peptide bond, that's why we call proteins polypeptides sometimes, because it's a peptide bond that forms between the amino acids. The growing polypeptide moves on to the new transfer RNA. Okay, so while this is going to be joined onto here, the three of them will then be on the transfer RNA that has the UUU on it. The ribosome is going to shift one codon. Okay, so it moves one codon to the right, bumping out this old transfer RNA. It'll be gone. Another transfer RNA comes, the peptide bond forms, and it keeps on shifting in order to add all the amino acids. When it reaches the stop codon, then it'll be triggered to come off of the transfer RNA and be finished translation. When the protein has been built, it'll either be released for use by the cell or transported elsewhere in the cell in the endoplasmic reticulum. Okay. The messenger RNA can be read by more than one ribosome at a time, so sometimes you'll see a bunch of them bound and reading. It has a really short lifetime though, so it'll be broken down by enzymes in the cytoplasm, and more will be made if more of that protein is needed. So, hi. I would love a sugar donut and a cranberry juice. Thank you. Sorry. They come in with a nighttime snack, so this is my nighttime snack. Oh, well, that was kind of funny. I think the lady thought I was talking to myself. Anyway, this will be a lot clearer to see when you watch the animations. There's a couple that are posted on the blog. Um, so again, just kind of get the idea of what happens. The amino acid is on one end of the transfer RNA. So the ribosome is going to start reading the messenger RNA, and when there's a certain codon, then the anticodon that matches with the transfer RNA, that transfer RNA is going to come in, it's going to bind, and then the other amino acids are going to be joined to its amino acid. It's going to shift, the ribosome is going to shift down a codon and read the next one, and so on and so on. So the animation is really good to watch for that. All right. The message. The sequence at the start, like I said before, is always AUG. Now, when you go to university, you're going to learn about differences between prokaryotes and eukaryotes. So in other words, what happens in bacteria and in us. There are a lot of extra little things that they talk about and extra little bonding processes, but we're just looking at the basics of how to read the code. And for the most part, that's pretty much the same. Okay, there are special things that happen with our messenger RNA, but that's too complicated for right now. You'll learn about it at university if you take biology there. Okay, so like we said, AUG is the start codon. So when you're trying to go through and actually transcribe and translate, when you get to the messenger RNA, after you've transcribed the DNA, then you're going to look and find the start codon. So you look along it until you find AUG. That's where the ribosome will start translating the message. I tend to underline or do the codons in triplets so that they're easier. So just like in this picture, if you go like this with all your codons, it's a lot easier when you're trying to translate it. You have a look, read your codons from your table. This is another version of the table. Does the same thing, but I find the other one's nicer. So then you can see your 3 letters and the name of the amino acid, or the 3-letter code of the amino acid. So we'll use the other one whenever it comes to your exam. Okay, when you come to the stop codon, then you don't write stop, but you stop making the protein. Okay, the ribosome itself will stop reading, it'll separate from the messenger RNA, and it'll it may go and read another messenger RNA. So the 3 stop codons UAA, UAG, and UGA, they're all indicated on your chart, so you don't have to memorize them. The only one that you have to remember is the start codon. Okay, that little link for transcribe and translate a gene, that link is also on the blog. It's just a little activity that you could do on your own or as a little review. Ms. Power might want to do that with you, but we're going to do one for practice right now. Okay, so the first thing that you're going to do is write the sequence of DNA strand that you would get if you replicated the DNA. So remember, all the A's go with T's, all the G's go with C's. That's what we have in DNA. So if you're replicating the DNA, and this is the template strand, then you have to make sure that you pair them up properly. So if you can pause the video and give them a couple minutes to try and do it, you should have this on your sheet. Alright, so if we did replication, then A's go with T's, G's go with C's. So every time I have an A, I put it with a T. And opposite. C's and G's. Sometimes you get a little misaligned because type is different size than how we write. So just make sure you keep track of them, otherwise you'll end up with the wrong protein. We don't want that. Okay, so we did T, T, A, C, A. That's an A. C, T, C, A, C, T, G, G, A, T, G, A. Wow, this is exciting, I'm sure, to listen to me. Alright, oops. So, first thing you do is figure out if it says DNA replication, then you just pair them up. Okay, A's goes with T, G's goes with C. Now, second part says, write the sequence of messenger RNA that you would get by transcription of this DNA. So if we started with the template strand, instead of replicating it, if we wanted to say what the messenger RNA was like, okay, so again, we're starting, this is your template strand, the top one. If I want to transcribe that top one, then A's go with U's. Okay, so G's go with C's. So this is, I need to move this out of the way. Messenger RNA is what we get when we transcribe, right? So the messenger RNA, if we were to transcribe this first original DNA, would be UCC, UCA, TG. Oh, sorry. I meant to get you to try and do it. So if you haven't already tried to do it, then you should pause. Okay. Okay. So again, C A A A's, T's go with A's, A's go with U's. So what do you notice about it? It's just like as though you were replicating the DNA, but instead of putting T's, you put U's. So for some people, if they didn't have the replicating DNA part, some people find it easier to write it as though they replicated the DNA and then change all the T's into U's. That's one trick. you're trying to transcribe the DNA. Oh, what did I do wrong? Can't have any T's in RNA. Okay, so all the A's go with U's. So the DNA, if this was your DNA strand and you replicated it, then the DNA strand that's made looks like a messenger RNA strand if it was transcribed. The only difference is instead of T's, it would be U's. Alright, the third part says determine the amino acid sequence of the protein that would be made from this. Okay, so I will let you pause, and if you're having some trouble, then you can play and I'll give you a little hint. Okay, so if you're having any trouble, oops, one of the first things you have to look for is, here's protein. Okay, first thing you need to do is look for that start codon. So we start looking from the left to the right, and we look for the codon for methionine, the start codon. So it doesn't matter, you don't look in threes right away, you look and you find it first. So remember, it's AUG. So we find, oh, there it is, AUG. So that codes for methionine. Okay, now I need to go back here for a second. Where is it? And I need to take this over here. Because I'm going to need it in a minute. Alright. I wonder if I can write that small. Sorry. Okay, so we're going to go in threes. Remember? There's the pen. Okay, mark them in threes. Alright, now we put a dash. We're going to find out what's the next one. CCC. Alright, let's find CCC. So we start here. CCC, this row, CCC is proline. So that's supposed to be a dash per row. Okay, next one is CAG. So same row, and we look for CAG. So you can pause again if you want to let them try and do the rest on their own, and then we can show them the answer. AGG is the next one. AGG is, where am I? AGG. Argin. GUC. GUC. Next one is valine. Okay, UUA. UUA. Leucine. CAC. CAC. Histine. Okay, UCA. UCA is Siri. CUG. CUG is Lucy. GAA. GAA is glutamine. And UGA. Oh, stop. Do I write stop? No. Okay, so this protein had 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. 10 amino acids in it. Okay, so you will be getting some more practice at this, and I may do up a slide with just one for you to practice, and she can, and Ms. Power can open it for you. But basically, that's the process. So if anybody wants the video of this, remember the files and stuff, you can bring a jump drive and ask Ms. Power if you can have a copy of it. So you have just transcribed and translated DNA. You have decoded the message. You should feel awesome. This is the kind of stuff that they do all the time in forensics, like on CSI and all of that. Very cool. These pictures you should have, I believe, in your little booklet as well. They're just showing the processes and where they happen. Different versions showing the same thing. Okay, so if you look and remember, transcription happens in the nucleus, where DNA is transcribed to give you messenger RNA, which is single-stranded and small. You've done this drawing before. Okay, it leaves the nucleus, goes out into the cytoplasm, and the ribosome is gonna go onto it to read it. Okay, ribosome attaches to read it. As it reads it, transfer RNA, so this one's good because it shows the little clover leaf, transfer RNA. The certain ones are brought that have the right anticodon, okay, carrying the proper amino acid, so it puts them in the right order. When the amino acids are all been added, and there'd be a stop codon that says, hey, we're done. Okay. This is another picture showing the same thing. Okay, it's just 3 different pictures, all showing processes of transcription and translation. So I think on your little lab, little mini lab that you're going to be doing, it's going to ask you to tell some differences between transcription and translation. And I think we actually did a question in the review for when we were doing the endocrine system. We did transcription versus translation as a practice review question. Okay, so you may want to go back to that again because you'll probably understand it a little bit better now, having done it in more detail. So for instance, where do they occur? Transcription occurs in the nucleus, translation occurs in the cytoplasm. You should know that they have different things involved. How are they in common? They're both required in order for protein synthesis, or both processes are needed in order to carry out gene expression or to carry out the action of a steroid hormone. There's lots of different ways that you can say they're similar, and other ways you can say they're different. So you might want to look back at that question that we had done together for review. Okay, this one I'm going to do in a separate video. So remember, you will be asked to